Morning, dear friends. Myself Abdullah Sherikar, working as assistant professor in the Department of Pharmacology at Sri Vile Kelavani Mandal's Institute of Pharmacy, Dhule. Today, we are going to study about what is the relation between the iron and hemoglobin. Learning outcomes. First, after completion of this lecture, we will able to understand the formation of the hemoglobin. Second, how the iron metabolism is occurred in our body. Third, how the absorption of the iron is takes place. How the cell transportation and storage of the iron is occur in our body. And last, how the hemoglobin is destructed by the cell. Formation of hemoglobin. As we are very well known that pluripotent stem cells are converted into the myeloid stem cells, and myeloid stem cells are converted into the colony forming unit erythroblast. These colony forming erythroblast, they are converted into the proerythroblast, and that is the starting or the beginning point for the formation of the hemoglobin. And this synthesis or the formation of hemoglobin is continued further to the reticulocytes also. The common form of the hemoglobin which is observed in adult is hemoglobin A, which is having the molecular weight 64,458. And this hemoglobin molecule is a combination of 2 alpha chain and 2 beta chains. The hemoglobin chains. The different type of the hemoglobin chains are present, out of which one is the delta, second one is the gamma, third one is the beta, and fourth one is the alpha. Alpha, beta, gamma and delta, these are the four different type of the chains are observed in the hemoglobin molecule. Synthesis of this hemoglobin molecule, the 2-succinyl CoA, which is coming from the Krebs cycle, is combined with the 2 molecule of the glycine, which forms the 4-pyrrole molecule. This 4-pyrrole is converted into the protoporphyrin 9, and when it combined with the Fe plus 2, the heme is synthesized. The heme is combined with the polypeptide, and the outcome is that the hemoglobin chain is with the alpha and beta, which is having the molecular weight 16,000, is synthesized or the prepared. This hemoglobin chain contains the 2 alpha and 2 beta chains, and this completely form a hemoglobin A, that is a common form of the hemoglobin in the adult. Each hemoglobin chain has heme prosthetic group, which has free iron atom. Each hemoglobin contains 4 different chains, 1 alpha, 2 alpha and 2 beta chains, and each hemoglobin chain has 1 iron atom. This is bind with loosely with the 1 molecule of the oxygen. So therefore the 4 molecules of the oxygen or the 8 oxygen atoms are transported by each hemoglobin molecules. Now the primary function of the hemoglobin is to combine with the oxygen in the lungs, and this combined oxygen is transported to the peripheral tissues through capillaries. Now the hemoglobin molecules actually the oxygen does not form a bond with the 2 positive bond of the iron atom. Instead, it form a loose and bond with the coordinated bond of the iron atom, and which is a loose and reversible bond. The peculiar characteristic of the hemoglobin molecule that it has a ability to combine with reversibly and loosely with the oxygen. Now the iron metabolism. Total quantity of the iron in the body is near about average 4 to 5 gram, out of which 65% is hemoglobin, 4% is myoglobin, 1% is heme compound, which is used for the intracellular oxidation, 0.1% is combined with the transferrin in the blood plasma, and 15 to 30% is stored in the reticuloendothelial system and liver parenchymal cells as a ferritin. Now the absorption of the iron from the intestinal tract, the liver cell, they start to produce the apotransferrin, and which is released into the bile, and through the bile duct it is coming into the duodenum. In the duodenum, the apotransferrin, which is a beta globin blood component, which combine with the free iron, hemoglobin and myoglobin, which is which result in the formation of the transferrin. And this transferrin is again combined with the epithelial cells by the pinocytosis process, and they entered into the blood plasma as a plasma transferrin, which contains the iron atom Fe+. Now the iron is absorbed from the all part of the small intestine. Second point, the loose binding of the transferrin with the iron, which causes the easy release of the iron in any tissue. Excess of the iron in the blood is deposited in the liver cell, that is the major portion, and the minor is stored in the reticuloendothelial cells of the bone marrow. The dietary iron source, majorly it is coming from the meat as a myoglobin. And the intestinal absorption of the iron, it is a slow process, that is few milligram per day. Now the blood and cell transportation as well as storage of the iron. The blood vessels contains the transferrin. The transferrin releases the free iron, and this releases free iron is transported to the plasma, crosses the plasma membrane and come into the cell cytoplasm, where it combines with the apoferritin. So the apoferritin, it is nothing but a cluster of the iron atoms, which is having the molecular weight near about 4,60,000, and this apoferritin is converted into the ferritin, which is considered as a storage iron. That means it contains the small amount of the iron. Second point, if you closely observe with the electron microscope, ferritin is observed as a small in size. The remaining free iron, which is coming into the cell cytoplasm, which is an insoluble form of the iron, and which is called as the hemosiderin, which is having larger size, which is seen as a larger particle or the clusters. When low plasma iron level is present, in that case, this whatever the ferritin, that is the storage iron present in the cytoplasm, it is breaks into the free iron and apoferritin. This free iron is going to be transported to the blood vessels, where it is combined with the transferrin, and it is called as the plasma transferrin. From plasma transferrin, the iron is transported to the low oxygen level, and in the bone marrow it is going to be bind with the erythroblast. The erythroblast, they causes the ingestion of this free iron by the endocytosis process, and from the erythroblast the transferrin helps in the transportation of the iron to the mitochondria, where it causes the synthesis of the, where it brings about the synthesis of the heme. Now the destruction of the hemoglobin, the RBC after bursting, they releases the hemoglobin. After that, there are different cells of the liver, say example, liver Kupffer cells, bone cells and spleen. They bring about the phagocytosis of this hemoglobin. That is nothing but there is a liberation of these phagosomes, and these phagosomes under the influence of the lysosome causes the breakdown of this hemoglobin into the heme and bile pigment. After few days or the hours, the macrophages releases the free iron, which is used for the synthesis of the, which is used for the synthesis of the new blood cells in the red bone marrow, and some part of it is stored in the liver as a ferritin. And the macrophages, again they bring about the uh, they bring causes the destruction of the porphyrin portion of the hemoglobin, and which result in the formation of the bilirubin, and this form bilirubin is transported to the blood. From the blood it is removed from the body with the help of the liver. Now the daily loss of the iron, in man they excrete 0.6 mg per day, mainly in the fecus, whereas in women they excrete near about half of the double of this man, that is nothing but the 1.3 mg per day. It is because of the menstrual loss of the blood. Now multiple choice questions. Molecules of the dash dash molecule of the oxygen, that is 8 oxygen atoms are transported by each hemoglobin molecules. Option A 4, B 8, C 5 and D 6. The correct answer is 4 molecules of oxygen. Common form of the hemoglobin in adult is hemoglobin B, hemoglobin C, hemoglobin A, hemoglobin D. And the correct answer is hemoglobin A. Dash dash is an insoluble iron in the cytoplasm. One is the transferrin, second option ferritin, C apoferritin, D hemosiderin. The correct answer is hemosiderin. Next, dash dash is a storage iron in the cell. A option transferrin, B ferritin, C hemosiderin, D apoferritin. Correct answer is ferritin. Next question, dash dash is a dietary iron source. A hemoglobin, B globin, C folic acid, D myoglobin. And the correct answer is myoglobin. These are some long answer questions that may ask in your examination. First, explain the synthesis of hemoglobin, explain the absorption and metabolism iron, explain the events involved in the cell transportation as well as the storage of the iron, explain the step involved in the hemoglobin. References: Primary Principle of the Anatomy and Physiology by Gerard Tortora, 13th edition. Second, Textbook of the Medical Physiology by Guyton and Hall, 11th.